Aminoglycosides as a class of antibiotic includes medications to treat aerobic gram-negative bacterial infections. They can also act synergistically with other antibiotics to treat gram-positive bacteria. Aminoglycoside antibiotics consists of amino sugars combined via glycosidic linkages. The amino sugars are combined via glycosidic linkages to dibasic aminocyclitol moiety, which is the basis of classification of aminoglycosides. The core structure of aminoglycosides has variety of amino and hydroxyl substitutions that influence the mechanism of action and bacterial susceptibility to these antibiotics. Entry of aminoglycosides in the bacterial cell is composed of 3 stages. First stage involves increase in the permeability of bacterial membrane. The second and third stages are energy dependent. The first stage involves electrostatic binding of the polycationic aminoglycoside to the negatively charged components of the bacterial membrane. It attaches to the phospholipids and teichoic acids of gram-positive organisms, and the phospholipids and lipopolysaccharide of gram-negative organisms, followed by displacement of magnesium ions. These magnesium ions are responsible for cross-bridging and stabilization of the lipid components of the bacterial membrane, and their removal leads to disruption of the outer membrane of bacteria. The damage to bacterial membrane leads to enhanced permeability, and initiation of aminoglycoside entrance. Upon entry into the cell, the drug reaches the cytoplasm of bacteria via energy-dependent electron transport-mediated process. These antibiotics bind to the 30S ribosome, thereby inhibiting bacterial protein synthesis. Inhibition of essential proteins leads to the death of bacteria and inhibits the reproduction process of bacteria. There are different mechanisms through which bacteria have produced resistance against aminoglycosides. There are certain enzymes in bacteria to overcome the effects of antibiotics. For example, beta-lactamase enzymes destroy the structure of penicillin. Similarly, there are enzymes which change the structure of aminoglycosides to make them ineffective. These enzymes are called aminoglycosides modifying enzymes, or AME. They have ability to acetylate and phosphorylate hydroxyl or amino groups of aminoglycosides. As a result, binding affinity of the drug is decreased. Some strains of bacteria contain 16S ribosomal RNA methyltransferases. These enzymes modify specific ribosomal RNA molecules in a manner that blocks aminoglycosides from effective binding to its target. In some opportunistic bacteria like Pseudomonas aeruginosa, the intrinsic resistance to aminoglycosides is due to efflux pump system. This system causes the exocytosis of the drug, and this feature is genetic or can be developed after some modifications and mutations. Aminoglycosides are rapidly cleared via urinary tract, that is why they are ideal drugs for the treatment of urinary tract infections. The drugs included in the aminoglycosides class include tobramycin, amikacin, neomycin, gentamicin, streptomycin. Modifications in the structure of these drugs are made to improve efficiency and decrease the side effects.